So next one is the plasma membrane. So cell wall, which is followed by the plasma membrane. So here the cell wall, it is the dead cells. Okay. So whereas the plasma membrane, it is the living cell, living material. So plasma membrane, it is a biological membrane. So which is present both in prokaryotic and eukaryotic cells, and it acts as a barrier between the outer and inner surface of a cell. 1 minute. Okay. So plasma membrane, it is a biological membrane and which is present both in prokaryotic and eukaryotic cells. So it acts as a barrier between the outer and inner surface of the cell. So plasma membrane is present as the outermost layer or envelope in animal cell, but is present beneath the cell wall in the plant cell as well as the most of the prokaryotic cells. So generally, in animal cells, cell wall is absent. So it is the outermost layer in the animal cell, but in plant cells, it is present after the cell wall, beneath the cell wall. So the plasma membrane, which is also referred to as the cytoplasmic membrane. So another name for this plasma membrane is plasma lemma or cytoplasmic membrane or biological membrane. So different names are there. So the plasma membrane, which also referred as a cytoplasmic membrane, is a biological membrane that encloses the contents of the cell, that is the protoplasm, and which separates it from the outer environment. So the cell or biomembrane has the extracellular phase, that is facing outer environment, and intracellular phase, that is the facing towards the cytoplasmic. So here the biomembrane means, so it it has both, that is out the side which is facing outside, it is called as the extracellular phase. So the phase, the side which is towards the cytoplasm, it is called as the intracellular phase. So the plasma membrane, which appears trilaminar, that is protein, phospholipid bilayer, protein, when viewed under the electron microscope. So generally it is biomembrane structure, and here the trilaminar. So it is protein, on either side protein is present, in the center phospholipid layer is, lipid bilayer is present. So it looks like a trilaminar. So cell membrane or plasma membrane, which separates the interior of the cells from the outside environment. So mainly composed of lipid bilayer, which including the cholesterol, so which maintain their fluidity under the various temperatures. So selectively permeable and helps in transport of substances with the help of proteins such as the integral and peripheral proteins. So again here two types of proteins, so integral proteins and peripheral proteins are present. So based on their position, proteins position. So involved in variety of cellular process such as the cell adhesion, ion conductivity and cell signaling. So provides mechanical strength to the cell. So first cell was discovered by Robert Hooke in 1665 using the microscope. The cell theory was proposed by, so we already discussed this, Swan and Sleeden and Swan, they proposed a cell theory in 1830. So according to them, so all living organisms are composed of one or more cells, and the cell is the most basic unit of the life, and all cells arise only from the pre-existing cells. Okay. So the lipid bilayer hypothesis was proposed in 1925 by the Gorter and Grendel. So the most accepted biological model of the cell membrane was given by Singer and Nicholson in 1972. That is the fluid mosaic model. So again here based on the, so different scientists, they proposed different models for the arrangement of this plasma membrane. But accepted one is the fluid mosaic model. So in 1972, Singer and Nicholson proposed the fluid mosaic model. So fluid means individual phospholipids and some proteins can move sideways laterally in such a, in each layer. Therefore, it is called as a fluid. So mosaic means range of different proteins which are resting on the surface on through the phospholipid layer, gives its mosaic appearance. Okay, so here the fluid is phospholipids act as a fluid and proteins act as a mosaic model. Okay, so here this is the fluid. So the icebergs like that. So these proteins are placed in this lipid layer, lipid bilayer. So here different types of protein, that is that the proteins which are present on the peripherals, peripheral proteins, and some proteins are there present inside, that is the integral proteins. So chemical composition of this plasma membrane. So cell membranes, they contain a variety of biological molecules, mainly lipids and proteins. So carbohydrates are present in very small amount, that is predominantly glycoproteins. So composition is not set, but constant changing for fluidity and changes in the environment. So plasma membrane generally they contains 40 to 45% lipids, 50 to 55% proteins, and 1 to 5% carbohydrates. So lipids. So the cell membrane consists of the three classes of amphipathic lipids, that is phospholipids, glycolipids, and sterols. So carbohydrates are present in very small amount, that is glycoproteins, and composition is, so here the lipids, phospholipids more than 55% and glycolipids 2% and cholesterol 40 to 45%. So phospholipids again two types, that is glycerophospholipids and sphingophospholipid. Again in glycerophospholipids, phosphatidylcholine, phosphatidylserine, and phosphatidyl ethanolamine, and linsocetal. So these are the main glycerophospholipids are present, and sphingolipids are sphingomyelin. So phospholipids are the most abundant lipids in the cell membrane. So again, phospholipids consist of two classes based upon the alcohol moiety, that is glycophospholipids and sphingophospholipid. So plasma membrane is asymmetric in nature, contains different types of phospholipids in the outer and inner leaflet, result in the increase in the fluidity. So outer leaflet, the phosphatidylcholine and ethanolamine and outer leaflet, and inner leaflet contains the phosphatidylserine, phosphatidylinositol, and sphingomyelin and the inner leaflet. So phospholipids are the amphipathic, that is molecules consist of the polar head and unsaturated fatty acid tail. So this is the structure of the phospholipid. It contains the head and tail. So head is hydrophilic and tail is hydrophobic. So unsaturation in the fatty acid chain prevents the close packing of the plasma membrane. So next to glycolipids. So glycolipids only accounts for 2% of the total lipids, and the fatty acids in the glycolipids usually contain even number of carbon atoms and typically between, that is 16 to 20. Next cholesterol. So it is cholesterol is normally found dispersed between the hydrophobic tail of the membrane of phospholipids. So cholesterol regulates the fluidity of the plasma membrane. And at high temperature, this cholesterol inhibits the movement of the phospholipid fatty acids and reduces the membrane fluidity. At a cold temperature, cholesterol interfere with the fatty acid chain interaction and acting as an antifreeze cholesterol and maintains the fluidity of the membrane. So fluidity of the lipid layer. So at low temperature, gel-like organization and polar head tightly packed, tails are regular and membrane thicker. At high temperature, fluid-like organization and polar head loosely packed and tails disordered and membrane thinner. Next one is a carbohydrate. So plasma membrane also contains carbohydrates, that is glycoproteins. So these carbohydrates are located on the surface of the cell where they recognize host cell and share the information. So viruses and other parasites bind to these receptors and cause an infection. So these are the proteins. The plasma membrane has large content of the proteins, typically 50% of the membrane value. So here the integral proteins are transmembrane proteins. So another name for this integral proteins are transmembrane proteins. So description, the span the membrane and have a hydrophilic cytosolic domain, so which interact with the internal molecules, and hydrophobic membrane spanning domain that anchors it within the cell membrane. So main here ionic channels, proton pumps, G protein and coupled receptors. Examples for this integral proteins. And lipid anchored proteins, example is G proteins. So covalently bound to the singular multiple lipid molecules. Next, peripheral proteins. So they are attached to the integral membrane protein and are associated with the peripheral region of the lipid bilayer. So these proteins tend to have only temporary interaction with the biological membrane. And once reacted, the molecule are dissociated to carry on its work in the cytoplasm. So example for some enzymes and some hormones. So based on the functions and position, so different types of proteins. So integral proteins and peripheral. So integral proteins are permanently attached to the membrane and embedded in the whole membrane and serve as a carrier proteins and channels and enzymes. Whereas a detergent should be used to remove the integral proteins. So glycoporine are the example of integral protein. Whereas the peripheral proteins, they are temporarily attached to the membrane and located on the inner or outer surface of the phospholipid bilayer and serve as the receptors and surface antigens. So peripheral proteins removed by the salt, pH changes. So erythrocyte spectrin are the example of peripheral protein. So ion channels, which allows inorganic ions such as the sodium, potassium, calcium, or chlorine to diffuse down their electrochemical gradient across the lipid bilayer. So ion channels, which also plays an important role in controlling the electric behavior of the cell. And G protein coupled receptor, it is a single polypeptide chain that crosses the lipid bilayer 7 times, responding to the signal molecule, that is a hormones and neurotransmitter. So G protein coupled receptors, which are used in the process such as the cell-to-cell signaling and regulation of the production of the CAMP and the regulation of the ion channels. So functions of the plasma membrane: protective, that forms the outermost boundary of the cells, and digestive, takes in food and excrete the waste products, and selectively permeability, which helps in the transport across the membrane, and contains cell surface receptors, example glycoprotein receptors, which are present in the RBCs, and cell adhesion molecules present on the plasma membrane of the certain cells and plays important role in the process of inflammation. Junctions, which helps in the formation of various types of junctions, adherents, anchoring, along with the help of cytoskeleton elements. So this is about the chemical composition of the cell wall and plasma membrane. In next class, we will discuss about the models of plasma membrane. Okay, so I will stop here. In next class, we will continue. So these topics are important.
Madam, thank you very much. Some of the participants, they left the meeting because of they are preparing their own material. So thank you very much. A wonderful session regarding of this plant envelopes, especially cell wall, composition of the cell wall, and the layers consist of the cell wall and the plasma membrane model, functions of the cell wall and function of the cell membrane or plasma membrane is most important. So thank you very much. Tomorrow we will meet, ma'am. Thank you.